In this experiment, our goal will be to determine the rate at which a 1.5% H2O2 solution decomposes when catalyzed by the enzyme catalase. In order to accomplish this, we will determine how much of the H2O2 has been consumed after 10, 30, 60, 90, 120, 180, and 360-second time intervals. Our hypothesis for this experiment is, if the enzyme catalase functions to speed up the process of breaking down H2O2 into oxygen and water, and we add catalase into several cups containing H2O2 for various time intervals, then the longer that the catalase is allowed to react with the H2O2, more H2O2 will be decomposed. Before beginning this experiment, it is important that all members of the group are dressed safely and appropriately for the lab due to the harmful chemicals that will be handled during this procedure. In this lab, wearing an apron, goggles, gloves, closed-toed shoes, and long hair tied back was necessary in order to protect the group members from the dangers that this lab presents.
In order to determine the rate in which H2O2 decomposes when catalyzed by the enzyme catalase, our group followed a procedure that was provided in the lab's instructions. We first obtained 7 new plastic cups and labeled them with the 7 time intervals being observed: 10, 30, 60, 90, 120, 180, and 360 seconds. 10 milliliters of H2O2 was then placed in each cup. 1 milliliter of Catalase extract was then added to each of the 7 cups and was allowed to react with the H2O2 for the amount of time that was written on the outside of each cup. A lab member was always ready to begin a timer as soon as the catalyst was added. Another lab member would then swirl the cup gently until the time had elapsed, and a pre-measured 10 milliliters of H2SO4 would be added, denaturing the enzyme and thus stopping the reaction. We repeated these steps 7 times, allowing the reactions to proceed for the 7 time intervals we were observing.
Some notable qualitative observations made in the first section of this procedure is that the enzyme catalase was cold and transparent. Also, whenever the catalase was added to the 10 milliliters of H2O2, bubbles started to form, indicating that a reaction was taking place. On the other hand, as soon as the H2SO4 was added, the bubbles stopped producing, indicating the reaction had been stopped.
We then collected 5 milliliter samples from the 7 resulting solutions in order to begin the titration portion of the procedure. Each 5 milliliter samples would be titrated with KMNO4. The titration would be stopped when a permanent pinkish-brown color change was exhibited. And then the total amount of KMNO4 added would then be calculated in order to determine how much H2O2 was catalyzed during the different time intervals. A baseline that tells us how much H2O2 was in the original sample was provided for us by our teacher. Using this baseline and subtracting the amount of KMNO4 added to each solution, we would be able to calculate how much H2O2 was catalyzed during each time interval. In order to determine how much KMNO4 we added to each 5 milliliter sample, we counted the number of drops that it took for the titration to be completed.
Throughout this experiment, we collected data regarding how much H2O2 was broken down by the enzyme catalase over 7 different time intervals. In order to calculate this, we first needed to determine how much KMNO4 was required to titrate each sample. The method that our group chose to determine this was to count the amount of drops that it took to titrate the resulting solution of each time interval. We were able to determine that on average 23 drops equaled 1 milliliter of KMNO4, and then we use stoichiometry to convert the data collected in drops to milliliters.
From the data collected over the course of this lab, we were able to calculate how much H2O2 is catalyzed during each time interval. After 10 seconds of H2O2 reacting with catalase, 1.43 milliliters of H2O2 was catalyzed into oxygen in water. After 30 seconds, 1.64 milliliters. After 60 seconds, 2.21 milliliters. After 90 seconds, 2 milliliters. After 120 seconds, 2.73 milliliters. After 180 seconds, 2.30 milliliters. And finally, after 360 seconds, 2.82 milliliters of H2O2 had been catalyzed. Graph 1.1 depicts the amount of H2O2 catalyzed over the 7 time intervals. The light blue upward sloping line is the trend line for our data, which shows that on average, the more time that the H2O2 is allowed to react with catalase, the more H2O2 is catalyzed.
After the experiment was completed and all materials had been cleaned, we then removed our gloves and aprons and began to discuss our results and the meaning of them. Our objective in this lab was to determine the impact that the enzyme catalase had on the rate at which a 1.5% H2O2 solution decomposes. In order to test that, we allowed H2O2 and catalase to react for 7 different time intervals and then determined how much H2O2 was catalyzed in each sample by performing 7 titrations.
We hypothesized that the longer that the catalase was allowed to react with the H2O2, the more H2O2 would be decomposed. Overall, our data supports this, as we found that on average, as the time of the reaction increases, so does the amount of H2O2 catalyzed. For the majority of our results, the more time that the solution had been allowed to react with the catalase, the less amount of KMNO4 was required to complete the titration. However, we did have 2 outliers, which were 90 seconds and 180 seconds. A possible explanation for why these 2 intervals did not follow the trend that the majority of our data followed is due to the human errors made during the experiment.
Possible sources of errors include inaccurate readings of the syringe, using the same syringe to collect all 7 5 milliliter samples of H2O2 to perform the titration on, leading to possible contamination, and counting drops of KMNO4 instead of measuring directly in milliliters. This was probably the greatest source of error due to the fact that we noticed the drops of KMNO4 coming out of the syringe changed sizes over the course of the experiment. Although we were able to determine that 1 milliliter of KMNO4 equals 23 drops, this number is just an average and could have led to flaws in the data collected. Ways to improve the validity of the experiment include making careful measurements and determining where the plunger seal ends, especially when measuring dark-colored substances such as KMNO4, using a new syringe for each sample that is being collected, and subtracting the final amount of KMNO4 in the syringe from the initial amount in order to determine how much was required to titrate each sample.